Good morning, good afternoon, everybody. We're going to start our seminar. And I'm happy to introduce, please, today's speaker is Marcial Escudero, who's originally from Sevilla and actually did his PhD in the Pablo de Olavide University. And after that, he got a Fulbright fellowship that brought him across the Atlantic to the Morton Arboretum. He followed that with the Marie Curie in the Chicago region and also together with the EBD. And in 2015, if I'm not mistaken, he got a Juan de la Cierva grant in the University of Sevilla, where he's still now as an associate professor. His expertise is around plant evolution and biogeography and all the topics that he's going to discuss in his charla today. So the floor is yours. Thank you.
Thank you. As Leticia told you, I'm going to talk today about chromosome evolution and how we can link this to diversification, to plant diversification. And we are going to use both a macroevolutionary and also a microevolutionary approach. So this is the outline of this talk. First, I will give you some background about chromosome evolution, and also I will tell you what are the holocentric chromosomes. This is in the title, this word. Then we will first I will tell you first about chromosome evolution across the whole genus Carex from the plant family Cyperaceae. Then we will talk a little bit about comparative genomics. We are going to compare genomes from the same species and also from closely related species and also from between distantly related species. And then we will see what patterns we can infer from that. And finally, I will tell you, we will zoom in and I will tell you a little bit about which microevolutionary patterns we can also see in two different groups of Carex species. And finally, I will give you a summary of the talk with the take-home messages. So the first question is, how does a karyotype evolve? So there are like different chromosome transitions that allow a karyotype to evolve. You can see here a polyploidy, aneuploidy, and fusion and fission. So polyploidy entails a whole genome duplication, so you double the chromosome number and also the genome size. This is much more frequent in plants than in animals. Then we have here aneuploidy. So in this kind of transition, you lose or gain one single chromosome. This is supposed to be like a evolutionary dead end from an evolutionary point of view. It's not very successful. And then we have the fission and fusion. So in this kind of transition, the genome doesn't change, the content of the genome doesn't change, but the number of chromosomes may change. So for example, if you have a fusion, you lose one chromosome, although the genome doesn't change. If you have a fission, you can gain one chromosome. And also we have the chromosomal inversion, in which in the same chromosome you have a fission, you change the order of the genes, and then you have a fusion. So, and why chromosome evolution is important? This is important because chromosome evolution may drive diversification. This can be indirectly. For example, you can have a geological or a geographical or ecological process that starts the speciation process. And then chromosome evolution may reinforce this speciation process. But also, chromosome evolution can be a direct driver of speciation. And there are two main models of chromosome speciation. We have the hybrid diffusion model, in which we have a strong selection against the heterozygotes for two chromosomal variants that we can have in one species. And then we have the suppressed recombination model, in which we don't have this strong selection against the heterozygotes, but we have a region in the genome in which there is no recombination, and typically we have an increased rate of nucleotide change, and sometimes we may even have like clusters of genes or super genes that are in this region where we don't have recombination, and this cluster of genes may be ecologically adapted, and this may promote speciation. So now I'm going to tell you what are the holocentric chromosomes. Here you have the typical monocentric chromosomes. These are the typical chromosomes that we have studied in secondary school, in which we have a single centromere. So these are the typical human chromosome. 80–85% of the eukaryotes have this kind of chromosome. So you have a fission in a monocentric chromosome. You may have a chromosome fragment without a centromere, and this fragment will be lost in cell division. So probably this individual that carry this chromosomal variant will have a very low fitness. In holocentric chromosome, we have centromeres across the whole chromosome. Then you have a fission. The resulting chromosomal variant, we have a normal fitness. This one will have just 2 pairs of chromosome, of extra chromosome, in comparison with the ancestral karyotype. So holocentric chromosome appear independently in 19 lineages of eukaryotes. And I highlight here the order Lepidoptera. I will tell you a little bit about them. And especially I will focus on genus Carex from the family Cyperaceae. So in holocentric lineages, chromosome evolution is typically by fission and fusion. So it's very common to find species with a small number of large chromosomes, or in the species with a large number of very tiny chromosomes, because you have a lot of fission rearrangements. So holocentric chromosomes have been related to the early terrestrial ecosystem colonization by plants and animals. And also we can see that currently in the species assemblage in high mountains, we can see a high richness of species with holocentric chromosome, because they tolerate very well UV radiation because of this special type of chromosomes. So I'm going to focus now on the microevolution in genus Carex. So this genus is very large. It's probably the third one with more species in the angiosperm. It has a worldwide distribution, but especially rich in the temperate areas of the northern hemisphere. Okay. So from this genus, we have a very comprehensive phylogeny. So we have a phylogeny of 1,400 species out of the total of the 2,000 species. And we know that diversification rates are very heterogeneous across the genus. You have lineages with a low diversification rate, the blue branches, and then you have other clades with high diversification rates in red or orange. So we look for chromosome number reports for the species that we have in the phylogeny that I showed you. So we were able to find over 3,000 chromosome reports from over 700 species, so around 40% of the genus. So here I show you the phylogeny. So in the tips, you have the chromosome numbers. The minimum chromosome number is a diploid chromosome number of 10, and the maximum is 124. And here, based on a very relatively simple Brownian motion model, I can show you how rates of chromosome evolution is also very heterogeneous, with branches in red, high rate of chromosome evolution, and then also many clades with blue branches, which is low rates of chromosome evolution. So in order to to properly model chromosome evolution in the phylogeny and also to be able to link this with diversification rates. We use this chromosome model that is kind of new from 2018. So in this model, we had a set of parameters to model chromosome evolution in the branches. So in this case, we have fission and fusion. So we can go from 20 chromosomes to 21, or in this case, fusion from 20 to 19 chromosomes. So in this model, we can also include a parameter to model polyploidy, but because we know this is kind of very infrequent or almost non-existent in this genus, in order to keep the model more simple, we remove the polyploidy parameter. But also we have also a set of parameters to model chromosome evolution at cladogenesis, when lineages are originating. So we have a parameter for no change in chromosome number, and then other two parameters for fission at cladogenesis and also fusion at cladogenesis. So we integrate all of these anagenetic parameters, the cladogenetic parameters, and also a background extinction rate across the phylogeny in this chromosome model, which is implemented in the platform RedBase. So as I told you before, chromosome evolution is kind of heterogeneous across the whole genus. So we know that there are clades probably with higher rates of chromosome evolution than other clades. So here in orange or here in red, we can imagine that here the chromosome evolution is much faster. So in order to have this into account also in this model, we include what is called like two hidden states, A and B. So and for each of these states, we have the same set of anagenetic and also cladogenetic parameters. We allow transition from this hidden state A and B and vice versa. And also we allow cladogenesis with a change in this hidden state. This is also important for this kind of model because they have somehow a very high rate of type 1 error, so you can infer that your trait is important for diversification. So including this kind of hidden trait state somehow is better to avoid this kind of type 1 error. So what we got was actually a part of the phylogeny that we will call A. So this is a hidden state. I don't really know what's this, in which chromosome evolution is kind of very, very fast. Like this is a huge rate of chromosome evolution, like 15 fission and 15 fusion per million years. This is like huge. And then we have another part of the phylogeny in which chromosome evolution is kind of zero. We don't have any transition. This is in the branches, in the anagenetic. This is the posterior distribution of the anagenetic parameters. In the clades, we obtain kind of two different behavior for chromosome evolution. We have a part of the phylogeny in which chromosomal cladogenesis is much more important than non-chromosomal cladogenesis, and another part of the phylogeny, the part B, in which again chromosomal cladogenesis is kind of near zero, and most of the speciation events are without change in chromosome number. So basically, in summary, we have a part of the phylogeny in state A in which chromosomal speciation is positive in comparison with non-chromosomal speciation, and another part of the phylogeny in which chromosomal speciation is kind of non-important when we compare to the chromosomal speciation. So I show you. So I show you here the reconstruction of chromosome number across the phylogeny. So high chromosome number in green, low chromosome number in purple, and also these two hidden states. We can see somehow a light, very tiny association between high chromosome number and the state A. So when we have chromosome number, we are in the state A in which chromosomal speciation is very important. But this association is not really that strong. So we can have other parts in the phylogeny in which we have low chromosome number, and we are in a state in which chromosomal speciation is very important. So now I'm going to tell you a little about the comparative genomics for this part of the talk. We have several data sets. First, we have a linkage map for an species named Cares scoparia that is from Eastern North America. We also have a whole genome from the same species, from Cares scoparia. We have a genome from a closely related species, Cares cristatella. Then we have another genome for a very distantly related species in the genus Carex. So here could be like Scoparia and cristatella. Here we can find Carex little dali, and the common ancestor between the three species near to the root of the phylogeny. And also we include in the analysis another genome from a closely related genus in the family Cyperaceae. So first I want to show you the comparison between the Carex scoparia genome. This is from an individual with an haploid chromosome number of 33 from Michigan in the United States. And we compare that with the linkage map of a cross between two populations of Carex scoparia, one with 33 chromosomes and the other one with 32 from Wisconsin and from Indiana. And we found a huge amount of rearrangements of fission and fusion, like 14, plus 4 inversion. So this is kind of a very high amount of chromosomal rearrangements, taking into account that we are talking about individuals from the same species. So this is surprising, but at the same time, if you remember, I told you that in the branches of the phylogeny, we have a very high rate of chromosome evolution. So here we will be actually seeing what is happening in a terminal branch of the phylogeny. So what's happening in a terminal branch of a species. So this is kind of congruent with what I showed you before. So when we go to the comparison between the species, here we have again Carex scoparia, and we compare that one to Carex cristatella. So they diverged like 2 million years ago, and we found now the double of the rearrangements than when we compare population from the same species, 39. When we compare to the species Carex little dali, that is more distantly related to this one, so we go from 2 million years of evolution to 23 million years. We go from 39 to 111 rearrangements. So somehow we have 10 times more time, but chromosome rearrangements increase like 2.5 times. And when we compare Carex little dali with the sister, not sister, but closely related genus, and species from a closely related genus, Rhynchospora breviscula. So the divergence time is 50 million years ago, and the number of chromosome rearrangements is a small increase. So it looks like when we go back into evolution, it's like we have less and less chromosomal rearrangements. It's not like a linear relationship. So it gets like flatter and flatter. Like, I mean, of course, when we go back in evolution, each time we have more chromosomal rearrangements, but this is somehow like a first linear and then flat relationship. So, and when we see visually, when we see this comparative genomics plot, I mean, the synteny of these genomes are kind of very conserved. If you think about all of these chromosomal rearrangements that I just told you, still after 50 million years of evolution, you can see like some kind of chromosome that stay the same across the whole evolution of the genus Carex. So this is kind of bizarre, no? When you have here the red, you have another blue here, a light blue around here. So something is happening there that, I mean, we see like more conserved synteny that we would expect. So I checked what's happening in the butterfly. So it's another holocentric group. So here there are things that are similar and other things that are kind of different. So here we have the silk moth, Bombyx mori, and then we have two butterfly genera, Melitea and Leptidea. Melitea and Leptidea are from the same superfamily, and Bombyx mori is from another superfamily. When you compare this genome, are kind of very similar. Then you have a lot of rearrangement here between two genera from the same superfamily, which is not expected. And then we have a bunch of genomes from the same genus. So what is happening in the butterflies, in the order Lepidoptera, there is somehow like an ancestral karyotype that is very conserved across the whole order. And then there are 8 lineages with a super high rate of chromosome evolution, and they have a very different karyotype in comparison to the ancestral karyotype. So that's why Melitea and Leptidea have a so different genome despite they are from the same superfamily. Okay, that's kind of different when we compare with Cyperese. We don't really have this kind of ancestral conserved karyotype genome. But what is kind of similar is that here you have again Leptidea sinapis from Northern Europe and Leptidea sinapis from Spain. And again, there are so many rearrangements within species, around 30 fission and fusion. And then when we compare with other closely related species, again, we somehow see this kind of relationship that get flatter when we go back into evolution. And what these authors infer is that the same fission events are happening in parallel in different branches of the phylogeny, or the same fusion events here, 6 and other 6 here, happen in parallel in 2 different branches of the phylogeny. So there is some kind of convergent evolution for fission and fusion. They also found that the genomic signature or the architecture of this fission and fusion in the genomes are areas of the genome with very high density of repeat DNA. So if we go back to the Carex genomes, so here in the x-axis we have distance from the fission and fusion point, and in the y-axis we have a density of different genomic features like genes or different repeat DNA, like centromere DNA, which is a satellite DNA, and different kind of chromosomes. And we find a significant relationship between the distance from the fission and fusion points to the density of these genomic features. So if we go in the areas close to the fission and fusion points, we have a high density of repeat DNA, and when we go to distant areas in the genome is where we found a high density of genes. So basically the model is very similar to the butterflies. Again, these repeat DNA are the genomic signature of these fission and fusion events. So here I show you like a typical genome from a monocentric plant. So this is the centromere, so an area that is very rich to satellite DNA. And then next to that area, you have also a high density of different transposons. And then as you go further in the 2 arms of the chromosome, you get a higher density of genes. Typically, when you have fission and fusion in monocentric lineages or monocentric species, typically they happen in the centromere. So you can go from a metacentric chromosome to 2 acrocentric chromosomes, vice versa. You go from 2 acrocentric chromosomes to 1 metacentric chromosome. In holocentric species, what we have is a cluster of repeat DNA, and then a cluster of repeat of genes, then a cluster of repeat DNA, and a cluster of genes, and so on, across the whole chromosome. Those clusters of repeat DNA is what is called the holocentromeres, and they are responsible for the segregation of the chromosome during the cell division, and also they are kind of responsible for these high rates of fission and fusion. So they are happening exactly in the part of the genome that you have high density of the repeat DNA. So this is the genome of Carex escoparia. So you have a ring for the gene density, then satellite DNA, and different kind of transposon. So if you go to each chromosome, you have here like 33, the number of places where you have this high density of repeat DNA is not, it's very limited. There are just several of them. So the targets for this fission and fusion are kind of very conserved. They are constrained in the genome. So that's why probably we are having the same fission and fusion event in different lineages of the phylogeny, and that's why we start to get this kind of flat relationship when we compare evolutionary time with the number of fission and fusion. So now I'm going to focus on the microevolution. I'm going to show you two examples. First, what we call the Carex levigata group. This is a clade in the genus Carex with 4 species, although one of them is polyphyletic, so we can consider that maybe there are 5. And we have a Ras-Set phylogeny, a very well-resolved phylogeography of the group. So we have here Carex binervis from Northern Europe with 74 chromosomes. Then Carex pavlovargasi from Northern Africa, also with 74 chromosomes. Carex camposi from Sierra Nevada with 72 chromosomes. The other lineage of Carex binervis from the Iberian Peninsula with 74 chromosomes. And then this large clade of Carex levigata. We have a huge rate of chromosome variation from 69 to 84. So we have, for example, here one clade here with a population from southern Portugal and from southern Spain with chromosome from 72 to 74. And this clade with population here from northern Portugal with a range of chromosomes from 72 to 74. So this clade here from central Portugal and northern Europe with a range of chromosomes from 70 to 74. And this last clade, which is from Portugal, southern Spain, and also northern Morocco, with a range of chromosomes from 73 up to even 84 chromosomes. So, I mean, of course, part of the genetic structure of the group, and also part of the explanation for the distribution of chromosome number is probably related to the glacial dynamics from the Pleistocene. But still, we can see that chromosome numbers are not randomly distributed across the distribution. We can see like a very clear pattern in which low chromosome numbers are in high latitudes or high elevations, and high chromosome numbers are in lowlands or in low latitudes. So they are somehow, there is like an order in the distribution of the chromosome. So we wonder whether or not they could be locally adapted across the distribution of these species, as chromosome number may represent different recombination rates and also different linkage groups. So we perform an analysis of landscape genomics called redundancy analysis, that you build like principal components. I show you here the principal component 1 and 2, based on these 4 bioclimatic variables: temperature, seasonality, minimum temperature of the coldest month, precipitation of the wettest quarter, and precipitation of the driest quarter. I show you here the distribution of the population of the species. Here you have Carex levigata with the highest distribution, and also here in the plot. Across the axis of the bio16. Here you have the Carex pavlovargasi across the axis of bio4. We have 2 clades for Carex binervis, but this species is polyphyletic, so actually this makes sense with the polyphily of this species. And then here you have several populations of Carex camposi from Sierra Nevada. So for the Ras-Set dataset, we had around 10,000 loci, but this kind of datasets are typically very gappy, so you have a lot of missing data. So we select 700 loci that were the most complete across the whole dataset. And we ran this analysis to see if there is a significant association between these loci and the different bioclimatic variables. And we found that there is a significant association between a bunch of them and the 4 bioclimatic variables that we test. But the interesting thing here is one of the loci that we include in analysis was a chromosome number, and actually this is significantly associated with the bioclimatic variable bio6, and actually very closely associated to other loci in the genome that are also associated in the same way to the bioclimatic variable. So we think that this could be some experimental support that there could be some kind of a local adaptation of the different karyotypes in this species. As I told you, they represent different recombination rates and also different linkage groups. And I want to show you, to finish the talk, I want to show you also another study. This one is in a species named Cares hilodi. This one is from southwestern Iberian Peninsula and from northern Morocco. And I did this study when I was a postdoctoral researcher here in the Doñana Biological Station. And so I think it's interesting to show you what I did here. So I'm showing now the result from one of the population. So this is a population. So with the last lab, we georeference all the individuals in the population. So this is the same population. So the individual goes following a temporal water stream, okay? So that's why they have this distribution, following the distribution of the water also. So this red individual here is the same that you have here, and the blue here, it will be this individual here. So this is the same population, okay? So we genotype all the individuals in the population with 34 microsatellites that they show variation in the species, but they don't show any kind of variation in the whole population. In addition, we select several individuals across the population and we genotype them with RAS-seq, so thousands of loci. And again, we were not able to find any kind of molecular variation in the whole population. And we were able to karyotype like around 100 individuals in the population. So most of them, the blue dot, have 70 chromosomes, and the meiotic division is very regular, like 35 bivalents, so 70 chromosomes. But then we were able to find several individuals with other karyotypes and also with irregularities in the meiotic division. So for example, here you have an individual with 70 chromosomes, but with irregularities in the meiosis. Here an individual with 69 chromosomes, this is the same thing here. And then in this, so this is the river, and here there is some kind of temporal wet meadow, and there are a bunch of individuals. So we find here also individuals with 68 chromosomes with irregularities, 68 chromosomes with a regular pairing of the chromosome, and also the 70 chromosomes with irregularities. And we follow here, we even find here another individual with 72 chromosomes. So I just want to show you here, so how fast is chromosome evolution in this kind of holocentric species. Besides, we don't find any kind of molecular variation. Still, we are able to find 4 different chromosome numbers and even 6 different karyotypes. So also, we can conclude here, so you can wonder what came first, molecular variation and then chromosome evolution came after that. So in this case, we can conclude that first were the chromosome mutation, and still we don't have any kind of molecular variation. So just a summary of the talk. So we can see how at macroevolutionary level, chromosome evolution is very, very fast, especially in some part of the phylogeny that we call state A. In that part of the phylogeny, chromosome evolution is also very important for speciation. So these chromosomal transitions are responsible for speciation. We also can conclude that there is not a linear relationship between evolutionary time and fission and number of fission and fusion. We get this flat relationship, and we hypothesize that there are hotspots for fission and fusion that are high density repeat DNA areas. And the same fission and fusion events may happen in parallel in different branches of the phylogeny. And finally, at microevolutionary level, I show you how the distribution of chromosome number across the distribution of this group of species is not random. They are somehow ordered across the distribution of the species. And we detect a signal of adaptive evolution in a bunch of loci, but the most important thing is that one of them was a chromosome number. So thank you very much for your attention, and also to the many collaborators that have been working with me in the example that I show you today, and also, of course, to the funding agencies. Thank you.
Thank you, Marcial. Oh, I see there's already one question.
Marcial, thanks very much. Fascinating. I wanted to ask first, because my question is very basic from my ignorance about this thing. So how quickly do these changes lead to reproductive isolation? So say in the Achnelkoya population, as soon as you get that chromosome change, does that mean that those individual plants are then reproductively isolated from the other plants in the same population?
So
or what happens?
So in the so I have been doing like artificial crosses between different individuals with different chromosome number. So one of the problems of the chromosomal speciation, or one of the highest critics about this, that once you have a new individual with a new set of chromosome number, if this one cannot interbreed with any of the individuals of the population, probably with a drift, it will be loose. No? The interesting thing here is you can have so the individual with different chromosome number can have a gene flow, but when you cross, when you cross two individuals with very similar chromosome number, you get like a very high rate of seed germination. Everything looks normal, but when the chromosome number or the number of rearrangements start to be like higher and higher and higher, then you start to get like lower seed germination. So in principle, they can interbreed normally, but not with the chromosome variants that are very distantly related. So we can imagine that all the population with different chromosome number can have gene flow between them, maybe indirectly. So I can interbreed with you and you with the other, but maybe some of the crosses are not allowed. So.
Well, thank you very much. That was very interesting, and well, coming from the animal world, very puzzling, no? And because this is shocking compared to what happens with birds, for example, that they are completely synthetic, there is very little variation. So what do you think are the factors that facilitate that in some groups there is a huge number of chromosomal rearrangements, and in other groups this is something very exceptional? In birds we can compare lineages that have diverged tens of millions of years and have very little changes, and you find them within a population. So what are the factors that you think can dictate?
Yeah, I don't know. So this is a very interesting question, but I don't really know why some lineages have high rate of chromosome evolution. Of course, I could tell you, if you have holocentric chromosome, you will have a high rate of chromosome evolution, but there are also lineages that they have monocentric chromosome and still they have a high rate of chromosome evolution. So, I have to be honest with you. I don't know why some groups, across the eukaryotes, why some groups may have a higher rate of chromosome evolution, and others they show like a super conserved. I don't know.
So thank you. It was really interesting. I was wondering something related with what Andy said. So you proved in the last example, but it was like more easy to understand for us maybe, you put that you didn't find any genetic variation, but you found in the same population different number of chromosomes. So how can you explain that? Is because the chromosomes are not expressing the variation, the genetic information that have, or how can you explain this?
Okay. So we have the macrocell data for the whole distribution of the species. I just showed you this population. There is usually there is a high genetic variation in Portugal, more or less high genetic variation in Morocco. But then when you go to the Spanish population in Seville and Huelva also, there are like less and less molecular variation. So in Huelva there is a little bit, and then in Seville there is nothing, as I show you. So first, probably there's some kind of a bottleneck or more or less long distance dispersal genetic bottleneck. So the individuals that were able to arrive to that part, they carry just a little bit of genetic variation. That's why we don't find any variation. Actually, this population there was like in the area like a fire like 20 years ago, and we knew the species from Portugal and also in Seville from Ronquillo, but we didn't find any population there in the field trips that we did. And after the fire, the population, I mean, we found this population and also the populations in the area, in the fire area. So this species needs open, I mean, they cannot be in the canopy of a closed forest. They need light. So that's why probably we found the population after the fire. So probably the individuals that we found in that population that I showed you, they have been there for 20 years, maybe. After the fire, probably they colonized that area. Before that, it was too close to be colonized. So the scene here is like, probably the individual that are right there was have very little genomic variation. So they can self-pollinate, and then all the seeds probably were like very similar to the original individual. I would say that from the time that population was colonized 20 years ago, probably some kind of, I mean, we had enough time for chromosome number mutations and karyotype evolution, but not for molecular variation. They could still have some genetic variation, but I didn't detect that. For most of the individuals in the population, we had this 34 microcell light that they have variation in the other population, but not in this one. And for several individuals, we did RASEC. So we test this with thousands of loci, and we didn't find anything. Still, some variation could be. But I mean, from my point of view, I think there was some kind of rearrangement in the last 20 years, or more than one. So this is happening like very, very fast.
More questions? Yeah.
Thank you, Marcial. Very, very interesting. So for those of us working with systems with very low rates of chromosomal rearrangements, of course, we are looking always for functional polymorphisms in genes affecting traits and fitness, but we never pay attention to this kind of genomic rearrangements. So does it mean that in these systems, gene mutation rates can be lower than in other plants? Basically because it seems that the system opted for chromosomal rearrangements for this huge amount of weak spots throughout the genome, rather than accumulating functional polymorphisms in genes. So in Arabidopsis, for example, flowering genes, important ones, you know, in the regulators of these genes, what we see is a huge amount of functional polymorphisms, and it seems it's the way, you know, to regulate the important pathways, you know, to colonize new environments. But this is not the case. So would you expect a lower gene mutation rates in Cyperaceae rather than other systems?
Okay, so it's an interesting question also. I would say that maybe because this special genome, that could be the case, that maybe most of the speciation are driven by all of these fast chromosome rearrangements rather than high rate of molecular divergence. Maybe so. I will have to see more about that, but it could be.
Hi. Well, great talk. So you showed that chromosome number is associated to environmental variables through RDA analysis. So, and well, we are so used to interpret adaptation based on variant, SNP variants and sequence variation. So what's the mechanism by which chromosome number is affecting adaptation?
So if I understood correctly, so why may have this distribution of the chromosome, no?
Right. So I'm understanding that the chromosome number by itself is causing adaptation. Is your interpretation? But I don't see the mechanism why. Because I mean,
there are two working hypotheses here, which are very related. So in holocentric lineages, not only in Cyperaceae, they have seen this in other lineages. Typically, so you have to, so normally in the typical chromosome, the segregation function and the recombination are separated. So the segregation happens because of the centromere, which is in a single point, and the recombination is happening in the arms of the chromosome. So here we have both things very mixed. So the solution in many lineages of holocentrics is to reduce the number of chiasmata to the minimum. So typically you find only one chiasma per homologous pair of homologous chromosome in the meiotic divisions. Okay. So basically, the recombination rate is directly proportional to the chromosome number, because having more chromosome itself allows more mixing, and because the chiasma will be also proportional to the number of chromosomes. So, and there is a lot of literature about so low recombination rate is a low evolutionary potential, and high recombination rate will be like a very high evolutionary potential. But if you have a high rate of recombination, you can mix genes that maybe they don't perform very well. So any of those strategies will be like better in one situation or in another. So the population with high rates of recombination will be in southern Iberian Peninsula, northern Morocco, and then the ones that colonize northern distribution or high elevation in mountain have lower recombination rate. So, and also the other working hypothesis, which is very related, chromosome number itself is it represents like linkage groups. If you have one more chromosome, that means that genes that are linked in one karyotype, they are not linked in another. So they could be also be adaptative. So that's the those are the hypotheses.
Through recombination rate.
Yeah.
So, thanks.
Thank you. Do we have more questions? Yeah.
Perhaps I missed the point, but did you measure any phenotypic traits so that you can associate some traits to the number of chromosomes? Okay,
so we are working now actually on that. We have we have now like several karyotypes in the greenhouse. We have a cross between them and also self crosses, and we are going to study different traits that we think could be important and see if we can relate that to the different karyotypes. But I didn't show anything. I mean, we don't have the data. There is not any obvious. I mean, there's anything that you can see like very obvious. Like, okay, this karyotype has this different color or something like that. Yeah, it has to be something quantitative or something like that. We are working on that.
But that might be an explanation of why you find within the same population different individuals with different number of chromosomes. That's an explanation could be that they have different number of chromosomes just because the number of chromosomes doesn't affect phenotype.
So, I mean, for example, in this here that you have this smaller chromosome, I can see that the individual here in the wet meadow that is here, the individual is smaller, but this is very speculative. So this could be because here there are less water or whatever. So, but yeah, we are working on that, and we are working with this in the greenhouse with the same condition and to see if we find something. But I don't know anything about that yet.
We have one question on YouTube. It's a question from Lorena. I'm gonna read it to you. She's wondering how useful are reference genome assemblies in species with such high chromosome rearrangement rates, and how this diversity can be characterized and integrated in species genome assemblies.
Can you read the question again?
So, she's wondering how useful are reference genome assemblies in species with such high chromosome rearrangement rates, and how this diversity can be characterized and integrated in species genome assemblies.
Okay, so, I mean, despite this high amount of chromosomal fission, I mean, we kind of expect, like, given the case, we see like a very conserved synteny. So there are like chromosomes that didn't change across more than 20 million years of evolution. So I would say, yeah, it's very helpful to have the whole genomes to do this kind of comparison. And the other question,
don't ask me, but the second part is how the diversity can be characterized and integrated in species genome assemblies.
So I guess, for example, in the thing that I told you that we are working now, that we will have more reference genomes from some of the species that we are working in the greenhouse, and we will also do some kind of a low coverage sequencing for all of the individuals that we will characterize with the phenotypes. So, and of course, we will be able to reference all of these sequence in the reference genomes, and we expect to find patterns. I think it's very helpful to have this kind of information.
Thank you. Are there more questions from the audience? Wait.
Are they deployed?
What?
Are they deployed?
Yeah.
The question is because I assume that being triploid or tetraploid, there are many redundancy can remove lots of limitations in the arrangements.
So polyploidy is kind of super rare in genus Carex. In other Cyperaceae, we find more polyploidy. There are like 3 genera in which this is relatively you have a normal rate, but in general, in Carex and other genera of Cyperaceae, this is very rare. So we find some polyploidy in some species in the tips of the phylogeny. Actually, there is a small lineage that is sister to the rest of Carex, and in that lineage, for example, we have found, we can find 6 and 12 chromosomes, haploid chromosome number 6 and 12, so this is polyploidy. But in general, there is no polyploidy, and based on the genome information across there are no deep polyploid events in genus Carex, that's for sure.
I think there was a question, yeah.
Yeah, just a quick curiosity. In the Athnilcoya map again, it seemed like really the aberrant individuals are not sort of randomly distributed. It seems like it's a non-random spatial pattern, right? Well, how would you interpret that? Is it, you think it's just some kind of local dispersal effect that not the Athnilcoya population, the map of the Athnilcoya?
Okay. Yeah, I see here also, because this is surprising to have so many chromosome numbers and karyotype in a single population, but I wouldn't say it's enough information to claim for a non-random distribution, but
the middle one looks.
But here, looks in the place that we have this, so we have the temporal stream. And here we have some kind of a wet prairie with a bunch of individuals. The place that I told you that there are like smaller individuals, and here we found like a higher percentage of individuals with a different chromosome number, but still, I mean, I don't want to conclude too much about that. One thing here also is, I mean, maybe that karyotype is more successful here. I don't know, but also by the time that one individual arrived there, this one can self-pollinate and colonize the rest of the prairie, and it will have the new karyotype. So that could also explain that's why there are more there.
Yeah,
I don't know if they are locally adapted.
It looks like 4 different colors there in total.
So
like
you
2.
have the orange. We have 68 chromosomes, but irregularities in the meiotic division. So depending on the individual, maybe there is a trivalent and a monovalent or tetravalent, something like that. So the number of chromosomes is the same, but there is an irregularity in the pairing of the chromosome in meiosis. So you have the orange, 3 orange. Then you have 1 yellow. Is the one with 68 chromosomes but regular pairing in the meiotic division. And then you have one kind of a pink, which is the typical chromosome number but with irregularities in the pairing of the chromosome during the meiotic divisions.
So now you went back to this. I was wondering, you were interpreting the karyotypes, the novel karyotypes, as recently arising in the same population, but that's only because you don't detect any variation in SNPs, right? Marker variation at all in the rest of the population. But in those assessments of genetic diversity, did you include these other individuals? With
for the microcell that was 34. I think it's it's a decent number of microcellulites. Those are the ones with highest variation across the distribution, but here there is none. And we genotype like most of the individuals with the microcellulite. And this, just to check, we got like several individuals across just a few, and to try. Maybe with RASI we are able to find something, and we didn't find anything. With the microcellulite, I can tell you that they are included for sure. With the RASI, I don't remember. Probably, I don't remember now, but probably I include, because I had already the information of the karyotype, so probably I include some individual with variation.
The other interpretation is that the population was founded from different sources with different chromosome number.
Yeah. So I mean, I have many chromosome counts across the distribution of the species. Not as many as in this population. We really focus on this one to see what we can find. So in Morocco, you have 34 chromosomes, and in Portugal, you have 72. In Huelva, you can find 72 and 70. And in the only population that we have 69, 68, as far as I know, is in this one. So I will go more for the hypothesis that one individual arrived and self-pollinate and colonize, and maybe at some point there was some kind of chromosome mutation and self-pollinate again or whatever. And I will go more for that hypothesis. Actually, so I visit, when I was a PhD student, I visit this population. So these are data from 2014. So I kind of visit this population the first time in 2005. That was exactly after the like 2 years after the fire or something like that. I don't remember if the fire was in 2004, 2003, but and the number of individuals was much smaller. So now there are many more individuals than before. For example, in this area that are very close here. So I mark the individuals back in that time. So here the vegetation grow again, and now there is not individual because they need like direct light. But it was like, I mean, maybe 100 individuals, something like that. Now there are like, but that time it was 400. I don't know now how many. And I count like 10 individuals. In that time, I found like, I think most of the individuals with 70, but if I count like 10 individuals, I found like 2 or 3 individuals with 72 chromosomes. And then when I start to count again chromosomes like several years later, I only find 1 individual with 72 right here. But I remember that in the beginning of the population, there was some individual with 72, but I was not able to find anyone there. So, I don't know.
Thank you. So we passed the hour, but I'm going to check if there's more questions in the audience. And if not, thank you again, Marcial, for a very interesting presentation.
Thank you.